Hello, good afternoon, everyone. This is Thomas speaking from Safeter. I'd like to welcome you to our webinar organized jointly with Danyel Biotech, who has just become distributor in Israel. I'm the director of business development at Safeter, and the first part of our presentation is going to be held by Anna Kaplan from Danyel Biotech, who is going to give you a quick introduction to the product portfolio and services. And then in the second part of the webinar, I will present our company and our tools for interpretation of NGS data. Our webinar is going to take about an hour, and there is also a section for questions, so feel free to paste your questions in the Q&A section here in Zoom, and we will discuss your questions either as we speak or at the end of our presentation. So Anna, now over to you.
Hello everyone, can you hear me?
Yes.
Hi, so I'm really happy to meet you again, Thomas, today, and present a bit of Danyel, but I have to somehow display my screen, right?
Yes, so you can share your screen with us. Also, for your information, our webinar is being recorded for our future reference. So I guess now you shall be able to share your screen with us.
1 second. No. Share screen. Share. Okay. So.
Okay, I see your screen.
Okay, great. Okay, so first, as we discussed, we have to present a bit about our company, Danyel Biotech. So we have a new collaboration with you guys since a month ago, even less maybe. So so far we've been known in the field of genomics, maybe less connected with analysis and bioinformatics side of it, but very much into hands-on and genomics and sequencing and other types of service, biological service. But let me talk about a little bit Danyel Biotech. So it was established in 1998 as a reorganization of Gamida Group. It's a well-known company in Israel. It's a company that actually consists of few small companies, and Danyel Biotech is one of them. So we actually work in the field of academics and also industry with biotech industry. And we have multiple companies, we represent multiple companies, and we provide not only sales, but also application support and also technical support for all the things that we sell and provide services. So this company is always happy to learn new technologies and be in the front of science. So we're trying to keep up with pace. And I think the companies that we represent never stop, and they give us lots of new things to learn and to conquer. So this is Danyel Biotech new office. It's actually 3 years old office in Rehovot, in the middle somewhere. And they, on the right side, you see our warehouse, new warehouse in Modi'in. And so we have lots of space for stuff to keep. Field of expertise. So as I said, we're really quite experienced in genomics field, from NGS to different other technologies in genomics. And also we have different other departments within the company, like protein, and the molecular and cell biology department, and also bioplastic takes care of different plastics and stuff, and also automation. And now they recently acquired a small company, like maybe not so small, but quite well known in Israel, that holds a medical division, and we also have a part in this. So the structure in short, so we have our CEO, Luli Gurevich, and he's the one that established the company. And then we have different departments, as I said. So this is our team. I'm from genomics team, advanced genomic division it's called. So we have a manager, department manager, Mariana. And Mariana, you on the call, I hope. Okay, and now we have 4 application and sales staff. So it's me, myself, Anna. We have Livnat, Shmulik, and Nurit, and we're all giving support and sales, and we're actively working in this field of genomics. Danyel Biotech represents quite big companies, as I said, among them Illumina, G Healthcare, and Hamilton is the big ones. And also smaller companies, as you can see here, is a quite nice portfolio. In genomics, we have 10 Genomics, and we have Takara Clone company, and we have Sophia Genetics also with us, and Verogen and Archer, and now, since very recently, VarSome. So one more nice thing about Danyel, that we have, especially thanks to our CEO, out of the box thinking. So you can see we are participating in conferences. This is a very known conference in Israel that takes place every 3 years. It's called Ilanit. And Danyel Biotech takes quite a very important place in this kind of conference. So Luli, this year, our CEO, managed to provide a special truck that you can see in the picture, that actually all the exhibition, all the things managed to put there and invite the participants. And we also organized a very nice seminar for students, for actually to class students, like for teenagers, to learn biology with a simple methods, and I think it was really successful. So that's us, Danyel Biotech, and now we will be happy to learn more about VarSome, and I think we can benefit together, because we bring in the know-how in more hands-on and sequencing, and you bring the know-how in analytics of this, and together I think it's a great partnership. That's what I think.
All right, Anna, thank you very much for a nice introduction. Now,
thank you.
I'm going to take over with screen sharing.
Yeah.
Share. So here is my screen. All right, so let's get started. So Safeter is a precision medicine and bioinformatics company. We are located, or we are headquartered in Switzerland, in Lausanne, and we are on the market since 2014. And we develop tools for processing and interpretation of NGS data for clinical purposes, although we also serve research customers. There are many challenges, obviously, in bringing NGS technology to the clinical practice. However, from our perspective as a precision medicine company and bioinformatics company, we are focusing on 3 challenges in particular. Challenge number 1, annotation data are fragmented and spread over many data resources. There are tens, perhaps even hundreds of data resources where you can go after a specific piece of annotation data. So this is a very, 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 very fragmented, very difficult to work with. Basically, the interpretation process can be seen as a puzzle game, right? I mean, to get a complete picture, you need to place all the little pieces in the right positions, right? Change number two, that this is a never-ending process. I mean, what was valid yesterday doesn't have to be valid tomorrow or the day after, right? Our understanding, you know, of genomics and our understanding of mechanism of diseases is becoming, you know, every day is becoming every day better, and so there's a constant need for, you know, re-evaluation of our, you know, and samples and classifications. And there is also a little standardization and consistency in terms of interpretation of pathogenicity, meaning there might be two laboratories working on the same variant and reaching a very different, you know, conclusions as a consequence of having access to a different annotation data sets. So these are the main three challenges, you know, we are, you know, focusing on here in our company. And I think you would agree that the challenge really is the interpretation part of the pipeline. Yeah? When it comes to the read alignment and variant calling, this is relatively easy. You know, the real challenge really lies in the tertiary part of the pipeline, the annotation and interpretation. So these are the reasons why we have created VarSome, the free and open platform. I guess at least some of you might have seen it before. It's free and open to anyone. And so what VarSome does is that it aggregates and cross-references publicly available data. So currently, VarSome counts with data resources from over 50 or even more providers. So on VarSome, you can find, you know, annotation data from ClinVar, from RepSeq, from Ensemble, somatic data resources such as ICGC, PMKB, Cosmic, Civic, expression profiles, list of drugs, list of clinical trials, annotation data for copy number variation, structural variation, clinical data resources for genes, phenotypes, and diseases, in silico predictions, conservation scores, and so on and so on. Over 50 data resources matched together. And we keep adding new ones on a regular basis. On top of that, we keep the data on VarSome always up to date. Yeah. So whenever there is an update in ClinVar or in GNOME AD, let's say we get it and we make it available, you know, on VarSome in a very short turnaround times. And we are also implementing guidelines for standardization of interpretation here. So on VarSome, for example, you can find the automated interpretation of pathogenicity based on ACMG guidelines. And now we are also implementing guidelines for somatic interpretation, such as AMP, NCCN, or ASCO, which the AMP is coming as we speak in the first quarter 2020. However, the free platform, it's not only about data integration and keeping the data up to date. It's also about you. It's about users, right? So on VarSome, we have currently over 200,000 users using the platform every day. And as a consequence of it, there is also a growing number of contributions coming from our global community. In other words, you can find, you know, on VarSome, you know, publication links, functional studies, classifications, or comments, you know, coming exclusively from our global community, which constitutes a unique layer of data which is nowhere else to be found, and it further enriches the knowledge base. So it's also about sharing your knowledge and about benefiting, you know, from the knowledge of others. On top of the free platform, we have built VarSome Clinical. So VarSome Clinical is already a professional paid service for interpretation of NGS data, starting from FastQ or VCF. So VarSome Clinical, it's clinically certified as an IVD device, as an in vitro diagnostic medical device, according to the requirements for medical devices, and so it can be used in clinical settings. VarSome Clinical is also HIPAA compliant, which is very relevant for US-based customers. And our company is also GDPR compliant, and we are also certified with ISO certificates for data quality and data security. With VarSome Clinical, you can start either from FastQ or VCF. And so first, you shall upload your sequencing data to our data center. You can either start from FastQ or VCF. When it comes to the FastQ, it shall be Illumina or MGI technology. And when it comes to the VCF, we can take any kind of VCF data on VCF files as long as the VCF file conforms to the standards for VCF files. So we can take a VCF file from PathBio, Thermo Fisher, Ion Torrent, and so on. With VarSome Clinical, you can process any kind of NGS data, be it a gene panel, either commercial, such as Illumina, AmpliSec, or Swift Biosciences, assays, or it can be even your custom in-house built gene panel, or exome, or genome. It's open for any kind of NGS data. Once you upload your data, you can run the pipeline. VarSome Clinical offers a wide range of pipelines covering, pretty much all the clinical use cases. So we have pipelines for germline samples, for somatic samples, for de novo variants in trios, for a pipeline for carrier risk screening in couples, a pipeline for segregation kind of studies, including a pipeline for a copy number variation and structural variation. And the latest addition to the pipeline portfolio is a pipeline for UMIs, for unique molecular identifiers, allowing the detection of low frequency variants typically found in somatic samples. So a wide range of pipelines. VarSome Clinical comes with a website interface, which means you don't need to install anything locally. Everything runs on our physical server in Switzerland, and you access it over the internet as if it was a regular website. That means that you don't need to worry about the underlying hardware infrastructure. You don't need to worry about scaling up your infrastructure as the number of samples grow. You don't need to worry about updates, security, and so on. We take care of everything, and you access it simply over the internet. And in the last step of the process, you can generate a clinical report, which can be fully customized, both on the appearance as well as data entry level. So you have a complete control over it. You can set up your own template, according to your branding policy and according to your specific workflow. This is something what we have discussed already. The challenge really lies in the interpretation part of the pipeline when it comes, to the decision, when you need to decide whether it's a pathogenic or non-pathogenic variant. So this is our main advantage here. VarSome Clinical, it's not a complete replacement for bioinformatics expertise. VarSome Clinical, it's a productivity tool allowing your bioinformatics team, to be more productive, more systematic, to focus the energy on where it is really needed. And it allows you, to deliver, quick results quickly. So here you can see, just a list of our clients. As a Swiss company, we serve a number of clients in pretty much all the European countries, including Israel. And we do have a number of customers also on the global scale in the US, in Latin America, in India, Taiwan, Singapore, Hong Kong, and so on. From the business model perspective, VarSome Clinical doesn't have a license. VarSome Clinical charges on a per sample basis. depending on the size of the sample. So when it comes to the FASTQ data, the price depends on the number of megabases in reads. And when it comes to the VCF, the price depends on the number of variants. There are basically two ways how you can use VarSome Clinical. You can use it as a standalone solution. So with the standalone solution, you can process any kind of NGS data, as I was saying before, gene panels, custom gene panels, commercial gene panels, exomes, genomes. The platform is open for any kind of NGS data, and we basically, charge you on a monthly basis, depending on the use of the platform. So this is the regular, this is nothing else than the regular, pay as you go model. And there is another option called, the bundled solution, which is effectively a form of prepayment. Yeah. So with the bundled solution, we bundle our products together with our partners. So we can bundle, let's say, Illumina, AmpliSec, panel. We can bundle it with VarSome Clinical for data interpretation and reporting. So this is basically a form of prepayment where you can prepay, a certain number of analyses according, to your specific needs. We also do a local installation. This is just for your information. We can also install VarSome Clinical on your own premises. However, this is something what makes sense only for a large diagnostic and sequencing centers. We do work with distributors in Israel. We collaborate with Danyel Biotech, who is also Illumina distributor. So Danyel Biotech, shall be your first point of contact, and they are also very well familiarized with our services and tools. So they shall be able, to also provide you a basic support. For more complex, questions, of course, you can always reach out to me or to us directly. All right, so this is my quick introduction. Now, I'd like to show you the real platform. However, first, I think I'd like to start with the free VarSome here. I guess you have seen it before, so I'm not going to spend much time here. I'd like to tell you a little bit more about ACMG based classification of pathogenicity. So in this case here, you can see that 6 criteria fired based on the evidence coming from all these 50 data resources. So we don't rely only on 1 or 2 or 5 data resources. Instead, first we do the large scale data integration. We bring all the data resources together, and then we leverage it for variant interpretation. Yeah, so this is a very robust approach. So in multiple, for multiple criteria here, we leverage multiple pieces of evidence. So in the case of PP5 criterion, for example, here, you can see that we consider, ClinVar entries, UniProt entries, as well as contributions from our global community. And this is the case for pretty much all the criteria. We always, consider as many pieces of evidence as possible in order to give you a complete picture. So it's very robust implementation of ACMG guidelines. It's also very transparent implementation of ACMG guidelines, because here it tells you in a great detail, in a plain English, why given criterion fired or didn't fire. So this is something what allows you to review each particular criterion or each particular situation and make your own judgment call according to your best knowledge. Not all the criteria can be fully automated. That's the other thing. Certain criteria such as BP2, BP5, PM3, BP4, these criteria require a patient-specific information such as family history or allele segregation information. In other words, information which is not available to us. So it's you in those cases who is supposed to make the judgment call and trigger additional criteria based on your best knowledge. And third, it's also very flexible implementation, because here it allows you to specify the transcript, which may affect the final verdict. And you can also specify, the strength of the evidence in line with ACMG guidelines. So this is, the core the mechanism how we classify variants. Now, on top of the free platform, we have built VarSome Clinical. So VarSome Clinical is already a professional service, allowing you to start either from FastQ or VCF, and at the end you get a clinical report. So first you shall upload your sequencing data, right? So here you can you can upload your sequencing data directly from your computer. There is also an API endpoint, application programming interface, allowing you to upload the data automatically. Actually, for every step in the analysis, there is an API endpoint allowing you to automate the whole process. So once you upload your sequencing data, you can proceed and you can start the pipeline. So here we shall specify number of details. We shall specify the pipeline, whether it's a single sample, family trio, couple for carrier risk screening, germline or somatic pipeline. Here we shall specify the assay. So here you can see all the assays available from Illumina, from AmpliSec, Illumina Focus panel, or commercial gene panels from Swift Biosciences, for BRCA gene, for myeloid, and so on. Or it can be even your own customized gene panel. So when it comes to the customized gene panels, we need the BET file from you, where you specify the target regions. And then you can specify the reference genome, for example, AG1938. And then on the left side part of the screen, you shall specify the sequencing data to FastQ files. You can provide sample ID, sample descriptions, and here you can also type in the phenotypes in line with HPO, the human phenotype ontology. So here just type in the patient phenotype here, patient diseases as well here, and later in the process you can use these terms for variant filtering and variant prioritization. And then you are ready to start the pipeline. So once the pipeline is finished, you get the results in the form of a variant table. So here I'm going to show you first a whole genome sample with almost 5 million variants. So this is a healthy genome, quite typical genome, and yet with almost 5 million variants. So variants here are sorted by pathogenicity, and for each variant here you can see the variant name, variant type, the classification of it, ACMG criteria, AGVS term, transcript, gene, exon number, mode of inheritance, function. In terms of function, VarSome Clinical annotates against all the transcripts available, so whether it's coding, intronic, splicing, variant, frameshift, you name it, zygosity, population frequency according to the ethnicity, allelic balance, percentage of reads supporting the given variant, and coverage, which is a link which actually you can click on and it takes you to the JBrowse view where you can see the graphical representation of reads. There is also IGV browser as an option, as an alternative to JBrowse. Around the variant table, you can see the annotation data from all these 50 data resources split in these tabs. So here you can see, the frequencies, allele frequencies, clinically relevant information for genes, all the transcripts, clinical data for phenotypes and diseases according to HPO, And then here beneath the variant table, here you can see again, the ACMG interpretation grid, very similar to the free platform. The region browser, where you can see all the variants within given gene with all the supporting pieces of evidence. So here, this variant, as you can see, there is a multiple evidence coming from GNOME AD, Uniprot, ClinVar, DBSNP, Caviar, Bravo, ICGZ, Cosmic, and so on and so on. ClinVar entries, Caviar entries, population frequencies from GNOME AD, from Bravo, somatic data resources such as ICGC, GDC, in silico predictions, conservation scores, publications, and also contributions from our global community. So these are the annotation data. Now, for example, this variant, you can see it's likely pathogenic, right? However, if you don't agree with the default verdict, for whatever reason, you may reclassify the variant. You may reclassify it internally, privately, as a benign or as a variant of uncertain significance, for example. Or you can set up a fully custom classifications, such as, variant important, from a pharmacogenomics perspective, or variant which needs to be discussed later. And then in turn, you can apply these classifications to your variants here, such as artifact, drug response, pharmacogenomics, and so on. And so that's why you can see here in this column here these marks. So these marks correspond to the custom classifications, and here you can always also see the name of the person who set up a particular classification here. So this is something what allows you, to build over time your private database of classifications. We can even take your manually created list of variants you maintain in your spreadsheet typically, and we can upload it, privately to your account with VarSome Clinical for the purposes of variant classifications. So this is really a powerful feature, allowing you to maintain your private database of classifications. What we also do is that we link your samples together. Yeah, so for example, here under the samples tab here, you can see the list of samples sharing the same variant. Yeah, so here you can see that this particular variant in the blue field has been found in a heterozygous form in two samples so far, in this analysis and in another whole genome sample analysis. Yeah. So this is something, what allows you, to go back and forth between your past cases and current cases, compare the patient phenotypes and diseases, and increase the diagnostic yield. VarSome Clinical, it's all about productivity, being systematic, and about increasing, first of all, about increasing the diagnostic yield. Yeah. So you can really, build your private database of samples as well as classifications. And over time, you will see that you can increase, that this is something that allows you, to increase over time the diagnostic yield for your patients. By the way, here you can see contributions also from our global community. Not for every variant, obviously, but for certain variants, we have, for example, here you can see a contribution from VarSome user in May 2019 classified. This is a pathogenic variant according to the ACMG guideline. ACMG guidelines, however, a VarSome user here says it's benign. So additional piece of evidence or additional clue allowing you to make your own judgment. Now here under the analysis actions menu, here you can access a QC reports, quality control reports. So for example, here you can access the basic QC report where you can see the sample details, the details of the analysis, such as data resources used for interpretation, pathogenicity, summarization according to the summarization of the primary and secondary part of the pipeline, the number of reads, number of duplicates, persons on target, and so on. Summarization according to the coverage, summarization according to the classification, the number of pathogenic variants, summarization according to each individual ACMG criterion, and in the end, summarization according to the function. This is the basic QC report. However, there are other more comprehensive reports apart from the basic QC report. So for example, here you can download also the BAM file, index BAM file, or you can also access to fast QC report. You can download the VCF file with full annotations, or you can also here, you can also access to coding coverage report. So the coding coverage report, this is an Excel sheet with a very detailed, coding coverage report broken down by individual genes, exon numbers, and sequencing depth. Yeah. So this is something that allows you, to troubleshoot. This is something that allows you, to identify regions with a low coverage or regions that have been skipped altogether. So this is something that allows you to troubleshoot, issues with sequencing or issues with assay design. And then there's a couple of other QC reports, such as a region list, coverage report, and so on. So now filters. So we have here 5 million variants here. So we obviously need filters. So here on the left side, here you can access a dynamic filters. So with dynamic filters, you can filter your variants based on number of criteria, such as the ACMG rules, based on frequency, based on pathogenicity, based on chromosomal position or ClinVar classification, based on zygosity function, gene list. You can apply a gene list and use it as a filter, or you can filter based on the call status, based on the allelic balance or coverage, a number of reads, in other words, and so on. So here I'm going to give you just a quick example. Let's say we want to filter out only rare variants with a frequency less than 1%. So now we are setting up a filter set. A filter set may consist of several filters, such as frequency filter, and the other one can be a filter for pathogenic and likely pathogenic variants. We give it a name, and now we can apply it to our whole genome sample here. Also, please know that everything functions really fast, right? We are working here with a whole genome sample with 5 million variants, and yet the filtering works super fast. So here you can see the results of the filtering based on the frequency. Over 4.5 million variants have been eliminated, and based on the pathogenicity filter for pathogenic and likely pathogenic variants, additional 339,000 variants are gone, and we are left with only 5 variants in the variant table. We can always, deactivate certain filters, or we can, reactivate them again, and the results, update dynamically here. Or we can go even one step further, and we can modify the whole filter set. We can include additional criterion for PP5, ACMG criterion with a supporting strength of evidence. So now, again, we are going to apply it, and now we are left with only 2 variants in the variant table here. Yeah. So these are the dynamic filters, which can be really easily modified, and you get the results, dynamically updated here. This is the first kind of filters, the dynamic filters. However, there is even more comprehensive, filtering called algorithmic filters. So with algorithmic filters, we can encode any kind of filtering scheme, according to your specific workflow. So these algorithmic filters can be fully customized and according, to your specific workflow. So here you can see several common examples of algorithmic filters. So with algorithmic filters, for example, you can do a de novo, you can call de novo variants in trios, or you can do a carrier risk screening for a couple, or you can perform a compound heterozygous kind of analysis or segregation kind of analysis, and so on. So these filters can be fully customized. And here you can see the results again. So here I'm going to show you this time an exome trio. So here you can see the original exome trio analysis with 290,000 variants, and with him based on sure select. This is, I think, Agilent exome. And here you can see a number of sub-analyses. So each sub-analyze, corresponds to the results of an algorithmic filter. So here, for example, you can see 260 de novo candidates in exome trio. So again, here you can see the results in the variant table. So this is the filtered view. So let's deactivate the filters first. So now, yeah, I see questions are coming. So I'll get to that in a while. So here you can see the 260 variants. Again, sorted by pathogenicity, and then of course you can lay over, the dynamic filters based on frequency, pathogenicity, or ACMG classification. Yeah. So you can really easily combine these customizable algorithmic filters with these already existing dynamic filters to narrow down the search space, which is super important, especially when it comes to larger data sets such as exomes and genomes. Now, the last missing piece is the reporting. So let me show you in the last few minutes how the reporting works. So, the process of the analysis, it's automated. However, when it comes to the reporting, it's your decision to decide which variant you are going to report or not, right? So once you take the decision to report it, you select it for export here, and then you can proceed and you can generate the clinical report. So the clinical report can be fully customized. You have a complete control over it, and basically what you need to do initially is to set up a template, and then you can use it all over again for all your future samples. So you can set up your own logo, you can provide your own styles, you can set up your own colors, fonts, and sizes. It can be also translated into any language. So that's the control over the appearance of it. And now you also have a control over the data to be included here. So here on the left side, here you can access, the widgets with annotation data. So here, for example, you can drag in the content area, and then you can drag in the information about the variant, then you can drag in information about the gene, information about clinical data resources for genes and phenotypes, list of drugs, list of clinical trials, and publications. So everything is editable here. You can provide your comment for the report. Here you can see the information for the variant, transcript, AGVS term, the intron number, exon number, frequency, zygosity. Here you can see the clinical data, list of phenotypes and diseases, mode of inheritance, additional clinical data, for phenotypes and diseases, list of drugs, list of clinical trials, and list of publications. So everything is editable, so you can easily remove publications, or you can add a new publication here at your convenience. And then once you are, happy with the report, you can download it here as a PDF file or as a doc file, which is the end of the process, basically. So VarSome Clinical, it's a complex platform. I've tried, to focus on the most important aspects of it to give you at least like a brief introduction to what's available here. With VarSome Clinical, you can process any kind of NGS data, be it a gene panel, exome, genome, including a pipeline for copy number variation, structural variation, UMIS. VarSome Clinical is clinically certified, as an in vitro diagnostics medical device according to the CE-IVD marking. There is no license. VarSome Clinical charges on a per sample basis, depending on the size of the sample, either in terms of megabases in reads, number of variants, and you can also prepay a certain number of analyses. And Danyel Biotech is our distributor in Israel. So now I guess we could take some questions now. So let me check. A Veret question from Veret Bar. Whether there is a way how to filter clinical trials, right? Yes. So now basically we are implementing like more advanced, filtering for clinical trials and for drugs in line with patient phenotypes and diseases. So currently we display all the clinical trials and all the drugs in the list, which is not probably the best way. So we are now kind of like improving that part. I mean, that's the way how we work. We always work very closely with our clients. We listen to them and we improve and we develop the platform based on the needs from our real customers. So no more questions here, Anna. Anna, would you like to bring something up? Have I forgotten to mention something important?
I have a question. Maybe I didn't hear it. You covered it, but this is something that can happen. Maybe if a analysis, if there was a sample with a failed analysis or a little amount of reads, and someone just wanted to check, if it's enough, and they finally discovers that it's not something technical. Is it possible to reload the same sample? How you see this issue as failed samples and reloading of the same samples?
Right. So first, when launching the analysis here, you can specify whether you want to call all the variants or only the variants which pass QC filters. Yeah. So here, for example, you can decide to call all the variants, and then you can filter out to ones which didn't pass the QC filters, for example. So again, here I'm going to show you. So with these algorithmic filters later, you can filter out the variants which fail QC filters, for example. So this is something we are going to call them, but you should not consider them for the clinical report, right? So, but still, it gives you additional clue. Maybe it tells you, maybe it tells you, but in this position there might be a significant variant. So it can be a kind of like an indication for you to resequence the sample and run it again. Here also. We regularly participate in the precision contest organized by FDA. So precision contest, it's kind of like a bioinformatics competition organized by FDA, the Food and Drug Administration Office in the US. So that's where we regularly participate, and here you can see the results, from the latest run. So sensitivity and precision of our pipelines, it's over 99.8% for SNPs and indels, and these are the results, from the latest runs on the FDA precision contest. So we don't develop aligners or callers. We use Sention callers and aligners, and then we validate our pipelines, either using the genome in a bottle samples, or we also participate in this kind of precision contests.
I have another question, Thomas. How often is the databases or annotation databases being updated?
Mm-hmm. Right, it's a very good question. Right, so here in the clinical platform, the results are frozen in the moment of the analysis, right? On the free platform, you always get the latest annotation data here. Yeah. So you can click on the variant here, and it takes you to the free platform where you can see the latest classification of it. Yeah. Because again, you know, as our understanding gets better pretty much every day, you know, that you shall regularly, you know, re-annotate your samples against, you know, the latest annotation data available. However, by default, you know, the results here are frozen, are fixed in the moment of the analysis, in line with what's known about the variant in the moment of the analysis. And so, for example, half a year later or a year later, you can come back, you know, to the same sample and you can re-annotate it against the latest data resources, and then you can filter out the differences only. Right. So we have a few minutes more, but there are no more questions coming from our audience. So yeah, so I would conclude VarSome Clinical. You know, it's a clinically certified platform allowing you to process any kind of NGS data, and thank you very much for your attention here. Anna, would you like to add some remarks, closing remarks? No remarks. All right then. So thank you very much for your attention. As I was saying, the webinar has been recorded, so I'll share the recording with you over email, and I will also send you some additional learning resources for your convenience. So thank you very much, and in case you have more questions, feel free to reach out either, you know, directly to me or to Anna Kaplan from Danyel Biotech. Thank you very much and have a good day. Bye-bye.
Thank you. Bye-bye.